Good morning, everyone. My name is Chanchal Gupta, and I am a pharmacology toxicology reviewer in the Office of Generic Drugs. Today, I am going to talk about safety evaluation of drug substance impurities in generics. In this presentation, I will talk about the key aspects involved in the safety evaluation of drug substance impurities. I will then describe how OGD-Pharm reviews the safety of drug substance impurities in generic products. This will be followed by some case studies which will highlight the key points and the common deficiencies occurring in the safety justifications of drug substance impurities. Before I go into details, I want to bring your attention to these 4 FDA guidances, which are the 4 pillars for the safety evaluation of impurities in drug substances. Now let's talk about the key principles involved in the safety evaluation of drug substance impurities. One of the foremost principles is that the safety of a generic drug product should be same as the reference listed drug. It is possible that the nature or the levels of drug substance impurities in a generic drug product are different than the RLD due to differences in the manufacturing processes. But the proposed levels of drug substance impurities should be qualified to ensure that the generic drug product is safe as the RLD. We will talk more about impurity qualification in the next few slides. The next important aspect is the maximum daily dose of the drug, which forms the basis for setting qualification threshold, that is the limit above which an impurity should be qualified for safety. And last but not the least, the context of use of the referencing application supported by the given DMF should be taken into consideration in the safety evaluation of impurities. Context of use includes the route of administration, duration of use, dosing regimen, and the target patient population of the drug product. Now I want to share how OGD-Pharm reviews the safety of impurities in drug substances. The safety evaluation of impurities is a two-stage process involving mutagenicity and general toxicity evaluation. For mutagenicity evaluation, OGD-Pharm relies on ICH-M7 guidance. Based on the duration of use of the drug product, the threshold of toxicological concern for mutagenicity is identified. The threshold of toxicological concern is an acceptable intake for a chemical that poses a negligible risk of carcinogenicity. TTC provides an acceptable limit of mutagenic impurities corresponding to a theoretical risk of 1 additional cancer per million. An important point to remember here is that TTC-based approaches described in ICH-M7 are based on lifetime exposure to mutagenic impurities. Therefore, the duration of use of the drug product depends on the total number of dosing days in the lifetime of a patient. For general toxicity evaluation of drug substance impurities, OGD-Pharm relies on ICH-Q3A guidance, which describes the qualification thresholds based on the maximum daily dose of the drug substance. Now let's go over the key aspects of mutagenicity evaluation. If the proposed level of an impurity exceeds the TTC, methods described in ICH-M7 are used to address the mutagenicity. The mutagenicity potential of an impurity can be predicted by using quantitative structure-activity relationship. Please note that the mutagenicity evaluation should be based on 2 complementary QSAR models, expert rule-based and statistical-based, as explained by Dr. Naomi Kroluk in her talk earlier today. If an appropriately conducted QSAR analysis predicts that an impurity is negative for bacterial mutagenicity, then no further testing is required. If the QSAR analysis identifies a structural alert for mutagenicity, an Ames assay can be performed to follow up on that structural alert. An appropriately conducted negative Ames assay would overrule any structure-based concerns for mutagenicity. However, if the Ames assay is positive, follow-up testing using in vitro and in vivo assays as described in ICH-M7 is warranted. During the review process, in some instances, such as cases where we have questions on the submitted QSAR justifications, or when there is lack of sufficient mutagenicity data, OGD-Pharm collaborates with our colleagues in the computational toxicology team who perform in-house QSAR analysis on the concerned impurities. OGD-Pharm also collaborates with different experts and working groups within CDER to navigate complex issues regarding safety evaluation of impurities. Now let's talk about general toxicity evaluation. General toxicity evaluation of an impurity is warranted if the proposed level exceeds the qualification threshold. As I mentioned previously, methods described in ICH-Q3A guidance are used to address the general toxicity of an impurity. General toxicity evaluation includes systemic toxicity and in some cases local toxicity evaluation depending on the intended route of administration of the drug product. General toxicity studies are needed in one species with a duration of 14 to 90 days depending on the duration of use of the drug product. The study design of the general toxicity study should consider the context of use of the drug product. The route of administration in the toxicity study should support the intended clinical route of administration. The doses tested in the toxicity study should provide adequate margin of exposure as compared to the proposed clinical exposure. General toxicity of an impurity can also be justified using published scientific literature. Please note that the full publications should be submitted to facilitate a comprehensive review and the context of use of the drug product should be taken into consideration. I will now present some case studies illustrating the key aspects I just discussed and highlight the common pitfalls observed in the submitted safety justifications for drug substance impurities. The first case demonstrates the use of in silico approaches for safety evaluation of impurities. Impurity A was a process-related impurity in drug substance. The proposed generic drug product supported by the given DMF was an oral tablet for chronic duration of use. The proposed level of impurity A exceeded the threshold of toxicological concern per ICH-M7 as well as the qualification threshold per ICH-Q3A. To justify the safety of impurity A at the proposed level, the DMF holder performed QSAR analysis for evaluation of mutagenicity as well as general toxicity, including systemic, reproductive, and developmental toxicity, as well as carcinogenicity. Upon review, OGD-Pharm determined that the DMF holder's QSAR analysis for mutagenicity evaluation was adequate. Impurity A was predicted to be negative for bacterial mutagenicity, and hence it could be controlled as non-mutagenic impurity. However, the use of QSAR analysis for general toxicity evaluation was not acceptable. Overall, impurity A was not adequately qualified for general toxicity and therefore the proposed level was not acceptable. This resulted in a major deficiency. OGD-Pharm recommended the DMF holder to either revise the limit of impurity A per the recommendations of ICH-Q3A. Alternatively, the DMF holder was recommended to conduct a general toxicity study of 90-day duration or submit published scientific literature if they chose to control impurity A at levels higher than the qualification threshold. In a nutshell, the take-home message from this case study is that QSAR analysis is not acceptable for general toxicity evaluation as QSAR is not validated for the endpoints of general toxicity studies. In addition, I also want to highlight the common deficiencies identified in the QSAR justifications for mutagenicity evaluation. These deficiencies include use of single QSAR model, use of models that are not fit for purpose for ICH-M7, and oftentimes full QSAR study reports being missing. The second case illustrates the consideration of duration of use of the drug product in setting impurity limits. In this case, impurity B was a potential mutagenic impurity in the drug substance. The proposed generic drug product was an emulsion foam for chronic duration of use. The DMF holder proposed that the maximum daily exposure level of impurity B was 120 microgram per day. The DMF holder claimed that the referencing drug product supported by the given DMF was applied for 2 weeks per the RLD label. Therefore, the DMF holder considered that the duration of use of the proposed product is acute and claimed that the proposed level of 120 microgram per day for impurity B was supported by ICH-M7. Upon review, OGD-Pharm talks determined that the DMF holder did not consider the total number of dosing days a patient would be treated in a lifetime for the given indication. OGD-Pharm talks determined that based on the clinical indication of the proposed drug product, it could be used intermittently multiple times throughout the lifetime of a patient. Hence, the proposed drug product belonged to greater than 10 years to lifetime duration category, and the TTC is 1.5 microgram per day. I also want to mention that the safety thresholds used by OGD-Pharm talks align with the review division of the RLD. Overall, the DMF holder did not consider the duration of use of the proposed product per ICH-M7, and therefore the proposed level of impurity B was not acceptable. In the regulatory recommendations, OGD-Pharm talks provided explanation about the chronic duration of use of the proposed product. OGD-Pharm talks recommended the DMF holder to either revise the limit of impurity B to the TTC or perform QSAR analysis to evaluate the mutagenicity of impurity B. If the QSAR results are positive, the DMF holder was recommended to either control impurity B at the TTC or alternatively conduct a mutagenicity assay to support a level higher than the TTC. The bottom line of this case study is that oftentimes the context of use of the drug product, that is the clinical indication and duration of use based on total number of dosing days, is not taken into consideration while setting impurity limits, which results in deficiency. Coming to case 3, this was an interesting case where a single DMF was shared by 2 ANDAs. The drug substance contained 2 degradation impurities, impurity C and D. This DMF was referenced by 2 ANDAs. ANDA 1 was an oral drug product with an MDD of 1.5 gram per day, whereas ANDA 2 was an intravenous drug product. The dosing regimen of the intravenous product consisted of a loading dose of 7 gram per day followed by a maintenance dose of 1.5 gram per day. The DMF holder controlled both impurities C and D at not more than 0.15% and did not provide any safety justification as such. OGD-Pharm talks first evaluated the mutagenicity of impurities C and D. We consulted the FDA's computational toxicology group who conducted an in-house QSAR analysis. Impurity C was predicted to be positive for bacterial mutagenicity, whereas impurity D was predicted to be negative for bacterial mutagenicity. Duration of use of both ANDA 1 and ANDA 2 were chronic. Therefore, the TTC to control mutagenic impurities is 1.5 microgram per day. Since this DMF is referenced by 2 ANDAs with different dosage forms and MDDs, the question arises that which MDD should be used to calculate the control limit for impurity C, which was predicted to be positive for mutagenicity. The answer is, for mutagenic impurities, the MDD, which is used chronically throughout the patient's lifetime, should be considered to calculate the control limit. This takes into account the cancer risk associated with cumulative exposure to mutagenic impurities over the lifetime. In this case, the chronic MDD for ANDA 1 and ANDA 2 is 1.5 gram per day. So our recommendations for impurity C were to control it at the TTC of 1.5 microgram per day and calculate the concentration limit by using the chronic MDT of 1.5 gram per day, or conduct an Ames assay to address the mutagenicity concern if impurity C is controlled at a level higher than the TTC. We then evaluated the general toxicity. Since the DMF is referenced by two ANDAs with different dosage forms and entities, I will again pose a similar question. Which MDD should be used to determine the qualification threshold of impurity D, which is non-mutagenic? The answer is, for non-mutagenic impurities, the maximum possible daily dose on any given day should be considered to set the control limit. This is because non-mutagenic impurities can have unique toxicity profiles and their effects can manifest by either acute or repeated exposure. In this case, the maximum possible daily dose is 7 grams per day, which is the loading dose of the intravenous drug product. Therefore, the qualification threshold for impurity D is not more than 0.05%. Our recommendations for impurity D were to control it at the qualification threshold recommended by ICH-Q3A using the maximum possible daily dose of 7 grams per day. Alternatively, if impurity D is controlled at a level higher than the QT, conduct a repeated dose toxicity study to address the general toxicity of impurity D, keeping in mind the context of use of the referencing product. This case describes a common situation encountered by OGD-Pharm tox when DMFs are shared between multiple ANDAs. DMF holders do not consider the MDD of all referencing drug products. In addition to the MDD, oftentimes duration of use, patient population, and route of administration are not considered in setting impurity limits and the submitted safety justifications. This is the last case study of my talk. It illustrates the use of inappropriate test concentrations in the Ames assay. In this case, there were two potential mutagenic impurities in the drug substance, impurities E and F. The proposed generic drug product was an oral tablet for chronic use. The proposed levels of impurities E and F exceeded the TTC for ICH-M7. The DMF holder submitted two independent Ames assays for impurities E and F. The DMF holder concluded that the impurities E and F were non-mutagenic based on negative results of the Ames assays. Upon review, OGD-Pharm tox found that impurity E resulted in 50% reduction in the background LON, indicating that it caused excessive cytotoxicity. In the other Ames assay, we found that impurity F was spiked in the drug substance and a neat impurity was not tested. Overall, the top concentrations of both impurities E and F were found to be approximately 500-fold lower than the OECD recommended maximum test concentration of 5,000 micrograms per plate. This inferred that the mutagenicity potential of impurities E and F was not adequately characterized in the submitted Ames assays. OGD-Pharm tox recommended the DMF holder to tighten the limits of impurities E and F to TTC for ICH-M7. Alternatively, if the impurities were controlled at levels higher than the TTC, OGD-Pharm tox provided additional recommendations. For impurity E, which caused excessive cytotoxicity in bacteria, the DMF holder was recommended to perform an alternative genotoxicity assay, such as an in vitro mammalian cell assay using neat impurity. For impurity F, the DMF holder was recommended to conduct an independent Ames assay using neat impurity tested up to the top concentration as described in OECD 471 guideline. This slide summarizes the common deficiencies observed in the Ames assay. Excessive cytotoxicity caused by test article, which leads to inadequate characterization of mutagenicity potential. Neat impurity is not used in the Ames assay. Oftentimes, Ames assays are not conducted per OECD 471 guideline and selection of top dose is not adequate. With this, I have reached the end of my talk. To summarize, OGD-Pharm Tox safety evaluation of drug substance impurities in generic drug product relies on FDA and ICH guidances, the maximum daily dose, and the context of use of the drug product are taken into consideration to determine the safety thresholds. Before I end, I want to emphasize on this really good resource, the FDA Good Under Submission Practices Guidance, which describes common recurring deficiencies related to impurities in DMFs. It also provides recommendations on ways to avoid these deficiencies to help reduce the number of review cycles for an ANDA to attain approval. These are the useful resources with hyperlinks for your reference in future. I want to thank the management of the Division of the Clinical Review and the Office of Generic Drugs, particularly my colleagues in the PharmTox team who helped me in the development of this presentation. I also want to thank the computational toxicology team within CDER with whom we frequently collaborate regarding QSAR analysis. Thank you for listening, and I will be back for the Q&A session a little bit later. Please type any questions you may have regarding this presentation into the Q&A part. If you have any questions on this presentation after this workshop is over, please send them to the email address shown on the screen by March 19th. Thank you, and please welcome Dr. Deborah Johnson from the Office of Pharmaceutical Quality, who will now deliver a talk on nitrosamines.